Welcome to section 17 of metabolism. In this section, we'll be discussing the metabolism of ammonia. Let's get started. This is the metabolic map provided in section 1 of metabolism. In this video, we're focusing on ammonia. The urea cycle, which can be seen right here, is an important part of ammonia metabolism. Let's zoom up on this pathway. This is a detailed figure of ammonia metabolism, which can be found in section 17 of metabolism. Notice that this figure shows both the alanine cycle and the urea cycle. Before we discuss either pathway, perhaps it would be most helpful if I provided a brief overview first. When proteins are catabolized, the amino acid group found on amino acids must be removed. In this figure, we can see an amino group shown on an amino acid right here. It's important to know that when the amino group is released, free ammonia is formed, which is a toxic substance. The toxic ammonia must go to the liver, where it can be converted into urea. This occurs through the urea cycle, and then the urea is removed from the body in the form of urine. Okay, with this in mind, let's dive into the pathways. I think it would be most helpful to discuss the alanine cycle first. So the alanine cycle, again, is shown right here. And notice that this is a coordinated series of reactions that occurs between the skeletal muscle and the liver. Through these reactions, the amino acids are able to be used as energy. Although not ideal, this cycle is important because it allows the body to break down muscle and utilize it as energy during periods of starvation. From the figure, we can see that the catabolism of amino acids results in the formation of alanine. Notice that the amino group from the amino acid right here was transferred to alanine right here. The alanine in the liver is converted to glucose via gluconeogenesis. The glucose can then go back to the muscle where it's metabolized back into alanine and the cycle begins again. Notice that in muscle tissue, when glutamate transfers its amino group to alanine right here, alpha ketoglutarate is formed. Conversely, alpha ketoglutarate in the liver right here can receive the amino group from alanine, which results in the formation of glutamate. In other words, these reactions are the reverse of one another. Also notice that these reactions require transamination enzymes. You can see these right here. And notice that these require vitamin B6 in order to function properly. You can see vitamin B6 right here at the bottom of the figure. Once glutamate is formed in the liver, it can release the amino group in the form of free ammonia. You can see that right here. And this occurs through the enzyme glutamate dehydrogenase. The free ammonia can then be converted to urea by the urea cycle. Okay, let's do a question. A 51-year-old homeless male presents to the emergency department with slurred speech. On exam, he has asterixis and is encephalopathic. Alcohol-related liver damage is suspected. Labs are significant for transaminitis, including an ALT of 200. This enzyme is responsible for transferring the amino group from alanine to what compound? From the question stem, notice that this patient has alcohol-related liver damage. Also notice that he has an elevated ALT. From the figure, notice that ALT is a transaminase enzyme right here, responsible for converting alanine to pyruvate. We're essentially asked what compound receives the amino group from alanine. As we can see from this figure, alpha ketoglutarate receives the amino group, which results in the formation of glutamate. So in answer to the question, alpha ketoglutarate receives the amino group. Okay, let's discuss the urea cycle now. The cycle begins with ammonia, carbon dioxide, and ATP. These molecules are combined by the enzyme carbamylyl phosphate synthetase 1 to form carbamylyl phosphate. Recall that carbamylyl phosphate synthetase 2 is an enzyme associated with pyrimidine synthesis, which is discussed in a separate video. Carbamylyl phosphate can then be combined with ornithine to form citrulline, and this occurs through the enzyme ornithine transcarbamylase. From the figure, you can see that this enzyme has a star next to it, and it's associated with a disorder called ornithine transcarbamylase deficiency. This is the most common urea cycle disorder, and like other urea cycle disorders, results in hyperammonemia. This is due to decreased urea cycle activity. The excess carbamylyl phosphate gets funneled to orotic acid, resulting in increased orotic acid. This idea ties in nicely with pyrimidine synthesis. This figure is from the section on purine and pyrimidine metabolism. Recall that carbamylyl phosphate is also used to produce orotic acid during the synthesis of pyrimidines. So if a patient has an ornithine transcarbamylase deficiency, carbamylyl phosphate accumulates and is funneled to orotic acid, resulting in increased orotic acid levels. Unlike orotic aciduria, which is due to a deficiency of UMP synthase, which you can see right here, ornithine transcarbamylase deficiency does not result in impaired pyrimidine synthesis. This means that while orotic acid levels will be high, symptoms of anemia will be absent. Ornithine transcarbamylase deficiency and orotic aciduria are often tested together because they both result in high levels of orotic acid, so it's important to be familiar with their differences. As I mentioned a minute ago, the urea cycle disorders, including ornithine transcarbamylase deficiency, result in hyperammonemia. So it's important to be familiar with the symptoms of hyperammonemia. Because the urea cycle occurs in the liver, liver disease or urea cycle disorders can both cause disruption of the urea cycle, which will result in hyperammonemia. The ammonia is most toxic to the brain, so it shouldn't be surprising that hyperammonemia results in neurological symptoms. These include slurred speech, asterixis, tremors, somnolence, and encephalopathy. The treatment will vary depending on the underlying cause, but lactulose is often used because it acidifies the gut and traps the ammonia in the form of ammonium, which can then be excreted in stool. So as I've shown here, lactulose facilitates the conversion of NH3 to NH4, which acidifies the gut, traps ammonia in the form of ammonium, and then is excreted in the stool. Rifaximin can also be used because it's an antibiotic that destroys ammonia-producing bacteria. Okay, let's discuss how hyperammonemia can cause neurological symptoms. This is a figure of the glutamate-glutamine cycle, which can be found in section 17 of Metabolism. Normally, glutamine in the presynaptic neuron, right here, is converted into glutamate. Glutamate is an excitatory neurotransmitter that enters the synaptic cleft and stimulates the postsynaptic neuron. The nearby astrocytes, which you can see right here, normally recycle glutamate. You can see glutamate entering the astrocyte right here. From here, it's converted into glutamine, which can then be used again for neurotransmission. However, when ammonia levels are high, the ammonia in the blood, which you can see right here, diffuses into the astrocyte and increases the conversion of glutamate to glutamine. So ammonia enters the astrocyte, which you can see right here, which facilitates the conversion of glutamate to glutamine. However, when ammonia is too high, this results in an overall increase in glutamine. Excess glutamine in the astrocyte acts as an osmotic agent and pulls water into the astrocyte. This inhibits the release of glutamine back into the presynaptic neuron. So decrease glutamine over here. Because there is less glutamine in the neuron, there will be less glutamate, which means the neurons are no longer able to effectively communicate. This mechanism is the basis for hyperammonemia, resulting in encephalopathy. Okay, let's continue discussing the urea cycle. Notice that the steps we've discussed so far have all occurred in the mitochondria. You can see the mitochondria is represented right here by this box. However, the next steps we'll discuss occur in the cytosol. It's important for you to know that the urea cycle is a pathway that requires both cytosolic and mitochondrial enzymes. So we left off with citrulline. Citrulline can be combined with aspartate to form arginosuccinate right here. Notice that this occurs by the enzyme arginosuccinate synthase. From here, arginosuccinate is converted into arginine by the enzyme arginosuccinate lyase. Finally, arginine is metabolized into ornithine and urea by the enzyme arginase. The urea can then travel to the kidneys, where it can be excreted in the urine, and the ornithine can be reused in the urea cycle. Okay, let's do a question. A 1-year-old boy is brought to the physician due to worsening episodes of somnolence. Labs are significant for hyperammonemia and increased orotic acid in the urine. A deficiency of an enzyme responsible for synthesizing citrulline is suspected. How would the activity of UMP synthase likely be altered in this patient? Hopefully from the question stem, you notice that this boy has hyperammonemia and increased orotic acid. This combination should immediately make you think of a urea cycle disorder. Because the question stem states that the deficient enzyme is normally responsible for synthesizing citrulline, we can deduce that this patient must have an ornithine transcarbamylase deficiency. From the figure, notice that ornithine transcarbamylase right here normally combines carbamyl phosphate and ornithine to generate citrulline. This defect results in carbamyl phosphate levels to rise. If we pull up the figure from the section on purine and pyrimidine metabolism, we can see that carbamyl phosphate right here is also used during the synthesis of pyrimidines. Notice that orotic acid is a metabolite of this pathway. This is why our patient has increased orotic acid in the urine. So the question asks, how will the activity of UMP synthase likely be altered? Because orotic acid is increased, we'd expect the activity of UMP synthase to also be increased.